features and functions of things that you find in prokaryotic cells. And then we went through and talked about the different membrane-bound organelles that you find in eukaryotic cells. We talked about their structure and also their function. And then we started the cytoskeleton last week, and we said that the cytoskeleton has, if we could just go back to this slide really quick, overall has these functions to maintain cell shape and support. It can also provide various types of cell movement and also be the tracks along which motor proteins move to move things within the cell or to help the cell itself move. We also said that the cytoskeleton consists of three different types of protein filaments. We said microtubules are one type, intermediate filaments and microfilaments. And what we've gone through so far are microtubules. We discussed their function, their structure, and then some of the structures that will contain them. So for example, we said that cilia and flagella both contain microtubules, and the bending of microtubules within cilia and flagella is responsible for the movement of both of those organelles. And then we talked about the fact that cilia and flagella have some differences in terms of their length, the numbers that a particular cell will have, and then also their beating pattern. We said while they have those differences, they have the same internal structure. So they both contain a set of microtubules that are ensheathed in a plasma membrane. And on the inside, you have what's called a 9+2 array, and it refers to a 9+2 array of microtubules. And what you have inside a cilia flagella are 9 what are called doublet microtubules around the circumference of the cilia or flagella, and then you have 2 microtubules running up the center of either a cilia or flagella. And then finally, we said at the base of cilia and flagella, you have what's called a basal body. And we said that a basal body was one example of what's called a microtubular organizing center. And the microtubules that emanate in both cilia and flagella emanate from the basal body that's at the base of cilia and flagella. Okay, the next type of cytoskeletal filament that we're talking about are intermediate filaments. And there's not too much that you have to know about intermediate filaments. The one structure that you have to know that's made up of intermediate filaments is the nuclear lamina. So if you remember back to when we talked about the nucleus of a eukaryotic cell, we said just inside the internuclear membrane was this fibrous meshwork that's called the nuclear lamina. And the nuclear lamina provides support to the nuclear envelope, and that nuclear lamina is made out of intermediate filaments. Another structure that contains them, but you don't have to remember for this class, if you've ever studied the anatomy of a neuron, neurons have these long tube-like extensions from their cell body that's called an axon. And what they do is they send an electrical signal down the axon, and then at the end of an axon you'll have another cell, and then it'll send that message to the cell it's communicating with. That long extension that's called an axon that neurons have contain intermediate filaments. So this is looking at the intermediate filaments inside an axon, and they provide that axon with strength, so that tubular extension, it provides them with strength. So that's really all you need to know about intermediate filaments. The last type of cytoskeletal filament that we'll talk about are actin filaments. They're also called microfilaments. And the protein that assembles to form this particular filament is called actin. So this is showing you one actin protein. And in this picture, one sphere is one actin molecule. And an actin an actin filament or a microfilament, the structure of it looks a lot like if you took 2 strings of pearls and you twisted them around each other. So if you took 2 strings of pearls and twisted them around each other, that's what an actin filament will look like. When you look inside different cell structures, actin filaments can be present as a single filament. They can also be present as a bundle of filaments, and then they can also be present as a meshwork of filaments. We'll talk about one structure that has a bundle of microfilaments, and then another structure that contains a meshwork of microfilaments. There's two major roles. That's light. For actin filaments. One is that they stabilize cell shape. And they also help an entire cell or parts of the cell to contract. And actually that's what is involved in muscle cell contraction in your body involves actin filaments, and we'll talk about how actin filaments interact with motor proteins to produce that contraction. And then on a lot of different cell types, the actin filaments will support the plasma membrane. So in a lot of different cell types, the actin filaments will support the plasma membrane. Okay, so one structure that contains actin filaments and actually contains a bundle of actin filaments is what's called a microvillus. And if you'll remember back to the very beginning of this topic, we talked about the fact that intestinal epithelial cells will have a bunch of finger-like projections that are called microvilli. And we said what those microvilli do is they increase the surface area of those cells and allow them to absorb more nutrients. Each of those microvilli have a bundle of actin filaments on the inside of them. Another structure that contains actin filaments, but this time it contains a meshwork of actin filaments, is called a lamellipodia. And so this is a picture of an actual cell. It looks a little bit like a dumpling. And what you're looking at here, the flat part here, the whole flat part is the lamellipodia. So when you're looking at this picture here, this whole part around here is the lamellipodia. And these are time-lapsed photos, so this cell is moving towards the floor on the slide, and it's moving because of that lamellipodia. So the lamellipodia is used for movement of the cell. And then this is also showing you a lamellipodia here, that large flat surface on that cell. Another structure that contains actin filaments are pseudopodia. And pseudopodia literally means false foot. And organisms called amoeba amoebas have pseudopodia. And this is another structure that is used for movement. So you'll see the cell extend in one direction for a little bit, and that cellular extension that exists for a little while is called a pseudopodia. And pseudopodia also contain a network of actin filaments. Okay, cell movement or cell motility is generally going to require the interaction between a cytoskeletal filament and something inside the cell called a motor protein. And there's different ways, we'll talk about them in a second, there's different ways that motor proteins can interact with cytoskeletal filaments to produce movement. But a motor protein is a protein that uses energy from ATP to propel itself along a polymer. And each of the cytoskeletal filaments is a polymer. So you can think of these motor proteins as walking along a polymer. So they're going to move along the polymer. So one way in which they can interact is at the very top here. So what you have here is a microtubule And in this type of interaction, the microtubule is going to stay still. And what happens is you have a motor protein here that looks kind of like an upside down Y. And so part of the motor protein attaches to the microtubule, and the other part attaches to some cargo that it's carrying within the cell. And what happens is the motor protein will walk along the microtubule and then carry the contents from one area of the cell to another. So that's one way things could be moved within a cell. Something you don't need to remember for this class, but microtubules also have a minus and a plus end. We didn't talk about that. But there's specific motor proteins that walk towards the positive end, and there's specific motor proteins that walk towards the negative end of a microtubule. The second interaction here is actually the type of interaction that happens in your skeletal muscle cells to produce contraction. So this is the type of interaction that occurs in skeletal muscle cells to produce muscle cell contraction. And what happens in this type of interaction is actually the motor protein stays still, and it's actually going to be the filament that moves. So what you have here are a group of motor proteins. Here you have an actin filament, and what happens is this would be called the head of the motor protein. The head of the motor protein grabs onto the actin filament and pulls on it, and that causes the actin filament to slide in one direction, in this case to the left. And what happens when your skeletal muscle cells contract is motor proteins called myosin are going to grab onto actin filaments, pull on them, and that causes the actin filaments to slide towards each other, and that causes your muscle cell to shorten when it contracts. So that's the type of interaction that happens between motor proteins and your skeletal muscle cells. And then at the very bottom, this is the type of interaction that actually happens in cilia and flagella. So we said that cilia and flagella both contain microtubules, and those microtubules are going to be connected to each other by motor proteins. And what happens in this type of interaction is actually both the motor protein and the microtubule are going to remain relatively stationary. So what happens, because they both remain relatively stationary, when the motor protein tries to walk along the microtubule, it causes the structure to bend. So it doesn't actually move very much, but it's going to cause the whole structure to bend. And that bending of the microtubules is what's going to cause a cilia and flagella to bend and then produce movement. So those are the three different types of interactions that can happen between motor proteins and cytoskeletal filaments to produce some type of movement for the cell. Do you guys have any questions about that? Okay. So that finishes up the cytoskeleton. The next topic that we have to talk about in cell structure is we're going to call protein sorting. And we're talking about eukaryotic cells still. And in protein sorting, what we're talking about is how does a cell decide where to put particular proteins. So every organelle in a eukaryotic cell has a particular set of proteins. The cytosol has a particular set of proteins. There's certain sets of proteins that are embedded in membranes. And the question in protein sorting is, how does a cell making a protein know to put that protein in a particular location? So how does it know that it needs to go to the mitochondria versus the ER or the Golgi apparatus? So the first place we're going to start is the fact that we said that ribosomes are the structures inside cells that produce proteins, that synthesize proteins. And ribosomes are made out of rRNA molecules, so several rRNAs, and then also a set of proteins. So ribosomes are these structures that consist of both rRNA molecules and protein. Okay, and then also what we're going to find out is that most proteins inside a eukaryotic cell are going to start to be synthesized in the cytosol. So they'll start to be synthesized in the cytosol, and then they're going to be directed after that to different areas of the cell. So they'll almost all start in the cytosol, and then after that they'll be directed to different areas. Just to show you two experiments that kind of get at how, or one thing that's involved in protein sorting in eukaryotic cells. So if you look at these two pictures here, what you have is you're basically staining the cells for a very specific protein. And so when you stain the cells, what happens in this way, what happens is the parts where the protein is located light up in the picture, and the parts that lack the protein are going to remain black. So basically what you have here is the nucleus of every cell lighting up because the protein is located in the nucleus. Okay, so this is 1, 2, 3, 4, 5, 6, 7, 8 different cells. That's the nucleus of each cell. And then what's not lighting up is the cytosol or cytoplasm of that cell. Then when you look at this picture, what's happening is the cytoplasm or cytosol is lighting up and the nucleus remains dark. So each black circle here represents a nucleus that lacks the protein, and then the cytosol or cytoplasm lights up because that's where the protein is located. And now the interesting thing between this picture and this picture is that it's actually the same protein, and there's only one amino acid difference between the two. So what they did was they took this protein, they changed the protein by one amino acid, and now rather than going to the nucleus, the protein stays in the cytosol. And the reason that that happens in this experiment is because this stretch of amino acids within the protein, so actually within the protein is a stretch of amino acids that tells the cell this protein is supposed to go to the nucleus. So you can think of this as a cellular address, like a mailing address that tells the cell it's supposed to go to the nucleus. And then if you change this address, it no longer goes to the nucleus anymore. It remains in the cytosol. So this stretch of amino acids within the protein is important for determining that that protein is supposed to go to the nucleus. And then on the next slide is a slightly different experiment. So here, so you're still staining the cell for a particular protein. And again, what lights up in the picture is the part of the cell that contains the protein. And again, the part that remains dark lacks the protein. So in the picture on the left here, the protein is present in the cytosol and the nucleus lacks the protein. And then this picture over here, all the protein is in the nucleus. So you have two cells here. And then the cytosol or cytoplasm lacks the protein. And what they did in this experiment is this is the normal protein. So the normal protein is present in the cytosol. And then all they did was they took this normal protein and they attached this sequence of amino acids onto that protein. And when they do that, the protein no longer stays in the cytosol like it's supposed to. Now it goes to the nucleus. So what that tells you is that that little short amino acid sequence is sufficient to get something to the nucleus. So basically by adding that amino acid sequence onto that protein, you told the cell that it's supposed to go to the nucleus, and that's where the cell puts it. So what that leads us to is one of the things that's involved in protein sorting are these things called sorting signals. And then one example of a sorting signal would be the one we just discussed. You don't have to memorize that as being like the specific amino acid sequence, but that would be a sorting signal that determines that the protein's supposed to go to the nucleus. But a sorting signal is a short amino acid sequence in a protein that directs the protein to its correct location. So you can think of sorting signals as sort of a cellular address, and they're actually a part of the protein itself, and it's just a short stretch within the protein, so a short sequence of amino acids within the protein that's going to direct the protein to its correct location within the cell. Okay, and the one example of a sorting signal that we'll study in this class is called the ER signal sequence. So the ER signal sequence is a specific example of a sorting signal. And proteins that have an ER signal sequence are obviously directed to the ER, so they're directed to the endoplasmic reticulum. Okay, so basically the way I'm going to split this up, and this is just to make it easier to study. It's not like there's exactly 3 pathways in the cell, but I'm going to break it down like it's 3 pathways. So basically this is going to be one pathway. This pathway starts in the cytosol and then ends in the cytosol. So the first pathway is going to start in the cytosol and then end in the cytosol. The second pathway that we're going to talk about is going to start in the cytosol. That's where you start. And then there's going to be a step where the protein is first sent to the ER. And then within this pathway, some of those proteins remain in the ER, but other proteins within this pathway are then going to be sent from the ER to the Golgi. And then some of those proteins will stay in the Golgi, whereas other proteins that are sent to the Golgi from the ER may end up being sent to the outside of the cell, the plasma membrane, or a lysosome or a vacuole. So this pathway the protein can end up in several different areas within the cell. Again, that would just depend on the protein that you're talking about. So if it's a protein that's supposed to function in the Golgi apparatus, it's going to be sent to the ER, and then from the ER the protein is sent to the Golgi, and then it remains in the Golgi. And then the third pathway that we're going to talk about is this one. So all the pathways start in the cytosol. So again, you start in the cytosol. And then in this pathway, you actually finish making the protein in the cytosol. And then after the protein's completely made, it gets sent to its correct location. In this case, that could be the nucleus, the mitochondria, chloroplast, or peroxisomes. Okay, so those are going to be the 3 separate pathways that we talk about. So if we go back to this first one, this is probably the simplest one. Proteins that lack a sorting signal, so proteins that lack any type of sorting signal are going to start to be made in the cytosol, then they're going to be finished being made in the cytosol, and then they remain in the cytosol. Okay, so proteins that lack a sorting signal are going to take this pathway. And that's not a mistake. Those are proteins that are supposed to act in the cytosol. So proteins that are supposed to act in the cytosol are going to lack a sorting signal so that they're made, finished, and then remain in the cytosol. Okay, and then what happens in this middle pathway is you're going to start to make the protein in the cytosol. And then you're actually going to stop. You're going to pause protein production. So you're going to pause translation. So you're going to stop making the protein. And then here you have the ribosome with the partially made protein. And it's going to travel to the ER, where it binds to the membrane of the ER. And then once it gets to the ER membrane, protein synthesis will continue. And either the protein is going to be synthesized into the inside of the ER, or it'll actually be synthesized into the ER membrane, depending on what type of protein it is. Okay, so you're going to start making in the cytosol, the protein in the cytosol. You're going to pause protein production, go to the ER, then protein production resumes, and then the protein is either synthesized into the ER or into the ER membrane. And then so some of the proteins in this pathway remain in the ER. So if it's a protein that's supposed to function in the ER, then once it's synthesized into the ER, it just stays there. Some of the proteins that are sent to the ER are supposed to function in the Golgi. So what happens is there's proteins in the Golgi that are first sent to the ER, and then the ER sends it to the Golgi in a vesicle. So that's how it would get from the ER to the Golgi apparatus. And then vesicles from the Golgi apparatus may take the protein to the outside of the cell if it's a protein that functions on the outside of the cell. It may take it to the plasma membrane if it's a protein that's supposed to be in the plasma membrane. There's also vesicles that go to lysosomes or vacuoles that carry proteins because that's where they function. Okay, but you can find proteins in all these regions in the Golgi apparatus, lysosomes, vacuoles, the plasma membrane, the outside of the cell. You can find proteins in all those areas that first are going to be directed to the ER. So they're sent to the ER first, and then after being sent to the ER, they're sent to those other locations depending on the protein. And those would all also require very specific signals. So there'd be very specific signals that tell the cell that it's supposed to go to the ER first and then to the Golgi apparatus. Okay, so what we're going to do you guys have any questions so far? Okay, so what we're going to talk about next are the specific steps that happen between the protein beginning to be made here, the protein production pausing, and then protein production resuming. So we're going to talk about how does it get directed to the ER. And so this has pretty much everything you need to know is on the slide there. And basically this is just showing you the steps. So these are supposed to be time-lapsed photos of the same ribosome. So the ribosome starts here. This shows you the ribosome disassembling after it's done producing the protein. And proteins that are sorted to the ER are going to have what's called an ER signal sequence. So what they're showing you in the picture there is the ribosome is going to start to produce the protein, and then the ER signal sequence emerges from the ribosome, and there's a specific complex in the cell called signal recognition particle. That's what SRP is in the picture there. Stands for signal recognition particle. And what SRP does is it recognizes the ER signal sequence. So SRP recognizes the ER signal sequence. So what happens is when the ER signal sequence emerges from the ribosome, SRP binds to it. So SRP recognizes it and binds to it. And then what happens is when SRP binds, it pauses protein production. So it's actually the binding of SRP that's going to pause protein production. And then what happens is that there's an actual a receptor in the membrane of the ER. So in this picture, this is supposed to be the membrane of the ER. And within the membrane is a protein that's called the SRP receptor. And the SRP receptor recognizes SRP. So after SRP binds to the ER signal sequence and pauses translation, then SRP binds to the SRP receptor, and that docks the complex with the ER membrane. So that's what brings the whole ribosome with the protein that it's making to the ER membrane. Then what happens is after SRP binds to the SRP receptor and docks the ribosome with the protein that it's making to the ER membrane, SRP actually leaves. And once so it on-binds and leaves. Once it on-binds and leaves, protein production resumes. So that's what they're showing you here. Protein production is going to resume. And the protein that this ribosome is making is actually synthesized into the ER. So that would be the end of that pathway, or that would be the end of this step. So this is basically everything that happens between protein production starting in the cytosol and then the protein finishing being made into the ER. You guys have any questions about the different steps, about what happens between the cytosol and the ER? Okay, one thing that I have to kind of define too is this first step in this pathway is called co-translational sorting. So that's why it says co-translational sorting to the ER. And co-translational sorting is sorting that happens while the protein is being produced. So, or let me say, the first step in the sorting process happens while the protein is being produced. And that's what we saw. We saw the protein starting. We saw the protein begin to be made, but then we paused protein production, and then it was first sent to the ER. Once it's been directed to the ER, then protein production continues. So that first step, sorting it to the ER, happens while the protein is being made. Okay, so that's going to be in contrast to this last pathway that we talk about. Most proteins that are found within the nucleus, the mitochondria, chloroplasts, and peroxisomes are going to undergo what's called post-translational sorting. And this is sorting. So just for us, maybe I should have explained this. So co-translational sorting. So remember, translation is the process where you produce proteins. So co-translational is while translation is happening. And then post-translational sorting is sorting that happens after translation is over. So that means after protein production. So this is sorting that happens after protein production. Okay, so what happens in this pathway is that the protein is going to start to be made in the cytosol. The ribosome finishes making the protein in the cytosol, and then the protein is going, after it's done being made, is sent to one of these locations. And this would also involve sorting signals too. But so the uptake of most proteins to the nucleus, mitochondria, chloroplasts, and peroxisomes is going to happen through post-translational sorting. Okay, do you guys have any questions about protein sorting or any of the three pathways? Okay, so the last thing that we have to talk about in cell structure and function are cell junctions. And these are going to be structures that hold cells together. Which is obviously going to be important for multicellular organisms. So multicellular organisms are going to have structures that hold cells together into tissues and then also hold them together. Sorry, this is the last thing. We're going to talk about cell junctions, then we'll talk about tissues. So second to last thing. So we're going to mainly focus on the types of cell junctions that you find in animals. So in terms of the ones you find in animals, there's three types of junctions that are found between cells. One is called anchoring junctions. And anchoring junctions will be involved in anchoring cells to each other. So anchoring junctions are involved in binding two cells to each other. They're also important in binding a cell to what's called the extracellular matrix. And you can think of the extracellular matrix as the scaffolding that cells grow on in animal cells, or sorry, in animals. So in animals, this would be the extracellular matrix would be something that's on the outside of cells, and cells are growing on this matrix. So there's anchoring junctions that will allow cells to adhere to that matrix that it's growing on. Tight junctions are another type of cell junction, and these seal cells together. So tight junctions seal cells together. And what they do is they prevent molecules and other things from leaking across a cell layer. So examples of cells that would be combined by tight junctions would be the epithelial cells that line your intestine. They would be connected to each other by tight junctions, and that would prevent things from leaking between cells in your intestinal tract. And then the last type are gap junctions. And what we're going to see the gap junctions do is they actually allow two cells to directly communicate with each other, because gap junctions are going to connect the cytoplasm of two cells. So what's going to happen is there's going to be a channel that forms in the membrane of one cell that links up with a channel in the membrane, the plasma membrane of another cell, and then those two channels line up and they form a pore that connects the cytoplasm or the cytosol of those two cells. And then there's certain things that are able to flow through gap junctions from one cell to another. As I'll point out later, one example of cells that have gap junctions that connect them are your cardiac muscle cells. So all your cardiac muscle cells are connected by gap junctions. And the signaling that happens through gap junctions in cardiac muscle cells is important for their ability to beat in unison together. So those will be important for that. So the next slide just shows you the different types of anchoring junctions that we're going to talk about. So the different types of anchoring junctions are going to be adherence junctions, desmosomes, hemidesmosomes, and focal adhesions. And the main difference between the different types is going to be whether it's connecting two cells to each other or whether it's connecting a cell to the extracellular matrix. And then also the protein that's used to connect the cell either to another cell or to the extracellular matrix. And then finally, they will also differ on how they're anchored on the inside of the cell. So a key component of anchoring junctions that are actually going to form the connections between cells are these integral membrane proteins. And integral membrane proteins are these proteins that are just embedded in the membrane of a cell. So they're one type of integral membrane protein. And two types of cell adhesion molecules. That are important. So sometimes this is abbreviated as CAMs, or cell adhesion molecules. One important type is called cadherins. So these are going to actually be the proteins that link either a cell to another cell or to the extracellular matrix as cadherins, and then proteins called integrins. Okay, so these are the four types of anchoring junctions that we're talking about. So if we start with adherence junctions, these are going to connect cells to each other via cadherins. So there'll be cadherin proteins in the membrane of one cell that are going to link up with cadherin proteins in an adjacent cell, and that's going to connect the two cells together. So via cadherins. And then in the cytosol, The anchoring junctions are bound to actin filaments. And then the next one that we'll talk about are desmosomes. And desmosomes connect cells to the extracellular matrix. Do you have a question?
Yeah. Where it says anchoring junctions are...
Oh, bound.
Bound.
Bound. Sorry. Sorry, bound. Yeah. Feel free to interrupt me if my spelling gets sloppy on the board, or if I speaking too fast or anything like that. Just feel free to stop me and ask for clarification. Sorry, desmosomes connect cells to each other via cadherins. So these also use cadherin proteins.
Do they connect to each other or the ECM?
Sorry, what's that?
Do they connect to each other or to the ECM?
Does what connect to the to each other at ECM?
Do they connect to each other as extracellular?
No, they connect two cells together,
Oh, okay.
not to the extracellular matrix.
Okay.
So these are going to connect cells to each other, so two cells together, also via cadherins. And then one of the differences between desmosomes and adherens junctions is that desmosomes are connected to intermediate filaments on the inside of the cell. And then the next one that you have are hemidesmosomes. And hemidesmosomes connect cells to the extracellular matrix through integrins. ECM just stands for extracellular matrix. So hemidesmosomes connect cells to the ECM via integrins, and hemidesmosomes interact with intermediate filaments. And then the last type are focal adhesions. And these also connect cells to the extracellular matrix. And then rather than being bound to intermediate filaments, they're bound to actin filaments. And they also connect cells to the ECM via integrins. And the previous slide just provides you, obviously, with a definition, pretty much what we wrote on the board. So this is showing you adherent junctions that are connect two cells together. So the purple proteins, the purple shapes in that picture are the cadherins. And so two cadherins from opposite cells are going to connect to each other. And then on the inside of the cell, the cadherins connect to actin filaments. This is showing you a desmosome. Sorry, I was looking for the one that went to desmosomes that are connect two cells together, again, via cadherins, but then on the inside of the cell, they're attached to intermediate filaments instead. Then you have hemidesmosomes and focal adhesions, both which are going to connect a cell to the extracellular matrix that it's growing on or living on. And then again, the difference between the two is whether it's going to be linked to the intermediate filaments or actin filaments on the inside of the cell. Okay. So just in terms of taking a closer look at the proteins that are involved. So again, this is just showing you in purple here are cadherin proteins. And so basically cadherin proteins you can think of having three separate parts. They have a part that's on the outside of the cell. They have a part that's embedded in the plasma membrane. And then they have a part that's sticking out on the inside of the cell. And so the extracellular part of the cadherin protein is going to bind to the extracellular part of a cadherin protein coming from another cell. And then what happens is the intracellular part of the cadherin protein links to, in this case, this is actin, links to actin filaments through an adapter protein. So that's kind of what cadherin junctions, picture of what it looks like. And this is just showing you integrins. So integrins would have this three domains. Again, they would have extracellular part that's in contact with the plasma membrane, and then a part that's on the outside of the cell. In the case of integrins, the outside part of the protein binds to some part of the extracellular matrix. So this is not something you have to remember for this class, but you may have heard of collagen before. Collagen is a part of your extracellular matrix. So this is just showing you this integrin binding to collagen. And then on the inside of the cell, again, the integrin is going to be linked to the cytoskeletal filament through an adapter protein. So those are just the two types of proteins that are involved in the different types of anchoring junctions that we just talked about. Okay, so the next type of cell junction that we are going to talk about are tight junctions. And I mentioned that these are going to hold two cells together, obviously very tightly, and they're going to prevent things from moving between two cells. And so the one example I gave you earlier, and that's also showing you here, is these would be the intestinal epithelial cells. And this would be the inside of your intestine where you're breaking down food molecules. And on the other side of the cell, you're eventually going to absorb those food molecules that will eventually move into the bloodstream. The tight junctions that are here prevent molecules from simply just passing through between two cells. And tight junctions are made by membrane proteins called occludin and claudin. That's on the slide here. So these are made by membrane proteins called occludin and claudin. And what they do is they form these strands in the plasma membrane that are shown here. And these strands of proteins are in adjacent cells. They bind to each other and they form a very tight seal between two cells. So that's what a tight junction would look like. And then the last type of cell junction that we're going to talk about is gap junctions. And I told you what happens in a gap junction is you actually connect the cytosol of two cells. So what happens in this case is these are going to be formed by a membrane protein, called connexin. Sorry, the membrane protein is called connexin with an I. So the membrane protein is called a connexin with an I. And what you have is 6 connexin proteins. You don't have to remember this part, but 6 connexin proteins are going to form what's called a connexon. And the connexon has a channel that's shown here. And then what happens is the connexon of one cell will line up with the connexon of an adjacent cell, and then that forms a little passageway that things can move between the two cells. And what happens, so I told you previously that your cardiac muscle cells are all connected to each other via gap junctions. And what happens in that case is the electrical signal that gets propagated across your heart that causes it to contract, that electrical signal moves through the gap junctions. And that way, what happens is if you stimulate the heart at one area, then the whole heart contracts in unison because that electrical signal that gets stimulated in one area is propagated throughout the heart through gap junctions, and then all the cells contract pretty much simultaneously. Okay. Do you guys have any questions about any of the cell junctions? There's also cell junctions in plant cells we're not going to talk about, and they're different, but that's just, we're not going to get into the different cell junctions that are in plant cells. So the last thing that we're going to talk about in this topic are the different types of tissues. And again, we're going to focus on tissues that are found in animals. So we're not going to talk about the different types of tissues that you find in plants again. So we said that tissues are the association of many cells of the same type. And as it says on the slide there, there's 6 basic processes that produce tissues and organs. I think the first 2 are pretty self-explanatory, cell division and cell growth. So cell division is when one cell divides and gives rise to 2 cells. So that's what cell division is. So a cell splits from being one cell and forms 2 cells. And cell growth is simply when a cell enlarges. And the enlargement of organs and tissues are the result of these 2 processes. So when tissues and organs enlarge, it's the result of cell division and cell growth. This process called differentiation is also important for producing tissues and organs. And what happens in differentiation is a cell becomes specialized. So in differentiation, a cell becomes specialized. So you start off with a nondescript cell, so a cell that isn't a particular cell type yet, and it specializes into a very particular cell type. So it starts off as basically you would call it as a stem cell, and then it would differentiate into, let's say, a cardiac muscle cell, or differentiate into a skin cell. So it would specialize, it would become specialized into a certain cell type. Another thing that's going to be important, and we'll see this later on when we discuss animal development at the very end of the course, is migration. That one's also pretty self-explanatory. Migration is when cells move from one part to another. And cell migration plays a really important role early on in embryonic development when your different tissues and organs, or sorry, not your different tissues and organs, but when these different cell layers within the embryo are forming. Another process that is important is called apoptosis. And apoptosis is a form of what's called programmed cell death. So this is actually a pathway that a cell can activate, or there's a signal that activates it. So the cell could itself activate this pathway, or there could be an extracellular signal that tells the cell to activate this pathway. And it's basically a cell suicide pathway. The cell kills itself. So when this gets activated internally, apoptosis can be activated into things in response to things like DNA damage. So if you're a multicellular organism and there's a cell in your body that has DNA damage that it can't fix, then for the betterment of the organism, that cell kills itself so that DNA damage isn't propagated further inside your body. And one thing that we'll study later on is that one thing that has to happen for cancer cells, or one thing that makes cancer cells special from normal cells, is that they're less likely to undergo apoptosis. So they'll be defective in pathways that activate this particular process. In terms of development, one place apoptosis plays an important role is in the formation of your fingers. So when you're initially in development, there's actually tissue between each of your digits. But what happens is all the cells that make up that tissue undergo apoptosis, and then that gives rise to your individual fingers later on, also your individual toes later on in development. And then obviously cell connections, which we just went through, are also important for producing tissues and organs because the cells have to obviously be connected to each other. Okay, so the last thing, so those are the 6 basic processes that produce the different types of tissues and organs that you're going to talk about, or that we're going to talk about. And the 4 major types of tissues found in animals, you probably are familiar with them. The first one is called epithelial tissue. These cells within epithelial tissue are going to be connected together through tight junctions. And epithelial tissue forms these continuous sheets. Okay, so epithelial tissue are going to be these cells that are connected together to form these sheets, and they're connected together through tight junctions. And epithelial tissue covers and forms the lining of all your internal and external body surfaces. So skin cells, your skin would be an example of epithelial tissue. The intestinal epithelial cells that line your intestine would be epithelial tissue. The cells that line, if you're female, if you have breast ducts, the cells that line those ducts are epithelial, is epithelial tissue. So epithelial tissue lines and covers all internal and external body surfaces. Another type of tissue is called connective tissue. And connective tissue provides support to the body and helps to connect different tissues to each other. So cartilage would be connective tissue. A third type of tissue that you're probably familiar with is nervous tissue. And nervous tissue is responsible for receiving, generating, and conducting electrical signals throughout the body. So a bunch of neurons together would be nervous tissue. And then the fourth major tissue type is muscle tissue. And muscle tissue generates force that facilitates movement. And muscle tissue would obviously be made up of a bunch of muscle cells. Oh, sorry. Generates force. Sorry, yeah, generates force that facilitates movement. Okay, do you guys have any questions about tissues or anything else that we've discussed today in terms of cell structure and function? Okay, I'm going to move on to, we're going to talk about viral structure and reproduction. And then next week we'll come back to the lecture questions for both this topic. And then I don't know if we'll get all the way through viral structure and reproduction today, but if we do, we'll do the problems next week. So that was just cell structure and function. And we're going to talk about viral structure, and then we'll also take this time to talk about its reproduction. We haven't talked about cellular reproduction yet. But one thing is that you probably are already aware of is that viruses are not composed of cells. So viruses are not composed of cells, and they're not considered living, so they're not considered living organisms. And so several reasons that they're not considered living organisms is that by themselves they do not use energy, which all living organisms do. They don't carry out their own metabolism, which all living organisms do. And they also do not maintain homeostasis, which again is something that all living organisms do. And then also on their own, they can't even reproduce. So we're going to see that in order for a virus to reproduce, it needs a living organism, it needs a cell in order to replicate itself. So as it says up there, viruses are non-living particles, but they do have genomes that are made out of nucleic acid. We'll see that the genome of a virus will differ depending on the type of virus that you're talking about. And we'll see that when we talk about their structure, it's going to include the genome, which is made out of some type of nucleic acid, and then there's going to be a protein coat that surrounds it. And then some viruses will also have what's called a viral envelope, which is a phospholipid bilayer that surrounds the capsid. The capsid is the protein coat. And then this is just showing you the various range of sizes in terms of different types of viruses. So viruses can vary in their size. And it's comparing the size of viruses to a bacterium and then also a eukaryotic cell. So viruses are smaller particles than cells are. As it says in the slide there, all viruses share some similarities. So they're all small. They all are going to rely on a living cell to replicate. So basically, a virus is going to have to infect a live cell, and then it's going to replicate inside that live cell, and then it has to escape from that live cell in some way. They also vary greatly. That's going to include both their structure, so both their protein coat can differ, their genome composition, so is it made out of DNA, is it made out of RNA, is it circular, linear, single, double-stranded? And then also they're going to vary in their host range. And I'll write the definition on the board for you in a second. But what you're talking about with host range is the number of different types of organisms the virus can infect. So if it can infect a lot of different types of organisms, then it has a wide range. But then there's other viruses that can only infect a certain specific species. And then even more than that, there are viruses that within that species can only infect a certain cell type. Okay, so we mentioned that all viruses have a protein coat, and that protein coat is called a capsid. And that capsid is going to surround the genome of the virus. And the capsid or protein coat can come in several different shapes. They can be helical. So this example, the tobacco mosaic virus, has a helical capsid. You can also have proteins that have polyhedral capsids. So this adenovirus here has a polyhedral capsid. Again, that's just a structure made out of protein. And then also, as it says in the slide there, many proteins that infect animal cells. An example would be influenza virus that causes the flu. Have a viral envelope enclosing the capsid. So what you're looking at here is these oval things here, and this virus is the capsid. The yellow thing in the middle is the genome. And then this blue circle with these, what they're calling spike glycoproteins coming out of it, that's the envelope that surrounds the whole virus. And that envelope is actually made out of a phospholipid bilayer that's actually derived from the cell that the virus infects. So what we're going to see is that when these certain viruses escape or are released from a cell, they basically get a little bit of the plasma membrane of the cell wrapped around them. And then that piece of plasma membrane also has viral proteins embedded in it. And then that surrounds, so that membrane actually came from the host cell that the virus infected. And then just one thing to mention down here, because it will come up later on in lab, is a bacteriophage. And this is simply the name of a virus that infects a bacterial cell. So bacteriophage is just a virus that infects a bacterial cell. Okay, so also the capsid and the envelope of a virus, one is important for protecting the genome of the virus, but it's also important in the ability of that viruses to infect a cell. So the capsid and envelope are also important for a virus's ability to infect a host cell. Okay, so one way in which viruses can differ is in the capsid. Another way in which they can differ is in what their genome is actually made out of. So this just gives you several different types of genomes of viruses. And so what you can see is that some viruses have RNA as their genome. And remember we said that the genome of all living organisms is DNA. So some viruses have an RNA genome. Other viruses have DNA genomes. And then you would differentiate between those two. Some are called DNA viruses. Other are called RNA viruses. And then you can also see that in some cases, like here, here, and here, the genome is double-stranded. So this is a double-stranded RNA genome, double-stranded DNA genome. And then also it can differ in the shape. So some are, sorry, not this one, but this one and this one are circular versus being linear. Okay. You guys have any questions so far? Okay. So the virus, the cell that a virus can infect is simply called a host cell. Then it says in the slide up there, if you're a species that can be infected by a virus, then you're called a host species. And then I told you that each virus is going to have what's called a host range. And the host range is the number of species and cell types that a virus can infect. Okay, so as I previously told you, some viruses will have a wide range of hosts, so they can infect several different types of species, whereas other viruses will have a very narrow range, some that only infect a single species. And then even further than that, there's certain viruses that only infect certain cell types within your body. One example of that would be HIV. Only infects a certain cell type within your body. You don't have to memorize anything from this next slide. It's just giving you an example of viruses in humans that infect specific cell types. And then also these viruses cause disease, some type of disease in humans. So those are all just specific examples of ones that infect particular cell types. You don't have to memorize any of them for this class. Do you guys have any questions? Okay, the last thing I want to do, because I want to get into the specifics of it. So I'm just going to give you a gist of what happens, and then next week we'll go through the specifics, because we won't make it all the way through the specifics if we start them now. But I previously told you that viruses cannot reproduce by themselves. So they're going to have to infect a cell, a living cell, in order to do that. And viruses, as we'll see, lack ribosomes. So we said ribosomes are the things that actually synthesize proteins. There are viral proteins, but all viral proteins are going to be made by ribosomes of the cell that the virus infects. It also lacks the enzymes necessary for protein synthesis in most, if not all, of the enzymes for nucleic acid replication. And what it's going to do when it doesn't have those enzymes is it's going to hijack the enzymes that the cell has and use them. Okay, so these are the viral reproductive. So the viral reproductive consists of 5 or 6 basic steps. And the details of the steps are going to, as it says in the bottom of the slide, are going to differ depending on the type of virus that you're talking about. What we're going to do is just take 2 specific examples. We're going to look at a bacteriophage called T7, and then we're also going to look at HIV, and we're going to look at the specific steps for those 2 viruses and compare them. So the basic steps here, they're pretty, I think, self-explanatory. So the first step is attachment, which means that the virus has to attach to the cell that it's going to infect. The virus is going to have to enter the cell. In some cases, we'll see for bacteriophage, all it does is inject the genome into the cell, and then the rest of the virus stays on the outside of the cell. In the case of HIV, what's going to happen is the HIV has an envelope, and the envelope of the HIV virus fuses with the plasma membrane, and that puts the capsid plus the genome on the inside of the cell. So first it's going to have to attach. Then the important part is getting the viral genome into the cell. And then in some cases, you're going to have integration. And what happens in integration is the viral genome is actually going to be incorporated into the host genome. So you're basically going to cut the host DNA, insert viral DNA, and then reattach it. And so you'll have the viral DNA integrated into the host DNA. And then you'll have synthesis of viral components. So this would include replicating the viral genome, producing the proteins that make up the capsid, and then also forming that envelope if the virus has an envelope. Then the virus is going to assemble. Some simple viruses can actually self-assemble without any help. The two viruses that we're going to talk about don't self-assemble. And then once the viruses have assembled, we're going to see that the virus is released. And we're going to see the case for T7 bacteriophage. We're going to see that release actually involves the cell breaking open. So the bacterial cells have a cell wall. And what's going to happen is the virus is going to break open the bacterial cell, and that's going to release the viruses that are inside. So you destroy the cell in that case. And then when we talk about HIV, we're going to see that release involves the budding off of a piece of the plasma membrane to the outside of the cell. And then that piece of membrane that buds off contains the capsid and the genome. And then that piece of membrane that surrounds the capsid and genome is now the viral envelope that surrounds that virus. So those are the basic steps of viral reproduction. And then we'll talk about the specifics next week for two specific viruses. Do you guys have any questions about the gist of what happens in each step?
So viruses don't hijack ribosomes, they just use them while they're inside particular cells?
Well, that's what we mean by hijack. Yeah, they use them while they're inside the cells.
Oh yeah, they don't like take them when they leave.
No, they don't take them when they leave. We'll see that, so yeah, they don't take, some viruses will contain more than just the genome on the inside of the capsid. So for HIV, it contains this enzyme called reverse transcriptase. And so that's a viral enzyme that the virus actually contains. And that enzyme is important for that virus because HIV has an RNA genome. It has a single-stranded RNA genome. So it needs an enzyme that basically copies that RNA, and actually it copies the RNA into DNA. And that's what reverse transcriptase does. It takes RNA and converts it into DNA, and then what happens with that is the DNA can then incorporate into the host cell genome. But yeah, in terms of ribosomes and most of the other machinery required for transcription, that's going to be hijacked from the cell. Any other questions? Okay, that's it for today. Just one, I don't think I put it in the announcements, but it is in the safety regulations for the lab manual. You do need to bring goggles this week for lab, so make sure we'll have extra pairs usually at the front, but make sure you bring your goggles to lab, and then your pre-lab, and then there's nothing else due this week for the class.